Hi everyone, Professor Stefero here, and this screencast is on osmosis. The yellow band across the horizon is the plasma membrane of a cell, and the molecules seen in the cytoplasm in the green and the extracellular fluid in the blue are water molecules. The movement of water molecules into and out of cells is critical to life, and although water is polar, it is small enough to pass through the plasma membrane. The diffusion of water across a selectively permeable membrane is called osmosis. It is a special kind of passive transport. Here we see plant cells losing water. That is, water is moving from a high concentration in the cytoplasm to a low concentration in the extracellular fluid until it reaches equilibrium on each side of the plasma membrane. So let's watch how osmosis works and then dig a little deeper to solidify the important concepts in understanding osmosis.
Diffusion is the net movement of molecules down a concentration gradient. This process allows small molecules, such as oxygen and carbon dioxide, to cross the plasma membrane. Most polar molecules, such as sugars and proteins, cannot freely cross this lipid membrane. Although water molecules are polar, they are small enough to pass through the membrane freely. This special case of diffusion that involves the movement of water molecules across a membrane is called osmosis. If a molecule, such as urea, is added to one side of a membrane, it will not be able to diffuse across the membrane because it is both large and polar. Because of its polar nature, it will interact with other polar molecules such as the water. This interaction reduces the number of free water molecules on the right-hand side. With fewer free water molecules on the right-hand side, there is now a net movement of water molecules down their concentration gradient to the side with the urea molecules. Because more water molecules are moving into this area than are leaving, the water level on the right side will rise. If the osmotic concentrations of two solutions are equal, the solutions are isotonic. However, when the solutions have unequal osmotic concentrations, the solution with the higher concentration of solutes is hypertonic, and the solution with the lower concentration of solutes is hypotonic.
All right, Professor Stefero again, and I want to dig a little deeper on some of the ideas and concepts that were presented in the previous video. First is to define a solution. A solution is equal to the amount of solvent plus the amount of solute. Let's say a 100 milliliter beaker, and the solvent is the solid blue material, we'll call it water, and the dots are the solute, and we'll just say that they're salt. A way to determine the concentration of the solution is to count the number of dots. That's what we'll do here. And if you count the dots relative to the amount of solution, we've got a 13% salt solution, solute to solvent. Thus we can say that the percent of water then that's making the solution is 87%. Solute is 13, solvent is 87%, and that's one of the things we have to know when we look at osmosis is how much water concentration is in a solution, because we end up following the water. All right, next let's take a look at this figure. The figure illustrates two solutions by a selectively permeable membrane. A membrane of this type will allow water to cross the membrane, but not the solute, salt in this case. Side A contains a 14% salt solution. Thus the amount of water or solvent in the solution is 86%. Side B contains an 8% salt solution. Thus the amount of water or solvent in the solution is 92%. The concentration gradient for water is not steep, but there is a difference between 86% and 92% that will lead to the osmosis of water across the membrane from B to A. So the amount of non-penetrating solute on either side of the membrane will determine the osmosis of water across that same membrane. And the U-tube experiment is another example of that. On the left, we have a solution with a lower solute concentration relative to the right side of the tube. Thus, water will move from the left to the right until the two solutions reach equilibrium. But if you look, right, the solute concentration doesn't change in the two solutions. It's the amount of water. The level on the left side has decreased and the level on the right side has increased. So the equilibrium gets reached because of the movement of water. And I can see what you're thinking. We have been following dots and they are not equal, but we're looking at the change in the water, and we can see that by the change in water level on each side of the membrane. Let's take a look at this example to see if maybe that will be more clear. In the picture, I've shown the solute as yellow dots and the solvent as solid blue. The 100 milliliter beaker on the left shows the initial concentration, which we might represent as 13 dots per 100 milliliters, or 13% solution. The beaker on the right is a 1000 milliliter beaker, and as a result, the concentration of that solution will be 13 dots divided by 1000 milliliters, and that will make it a 1.3% solution. So the amount of water relative to the amount of solute will change the concentration of any solution. So now if we go back to that original picture, we can see that the solute concentration appears to be different on each side of the membrane by the number of dots, but it's relative to the amount of the water due to osmosis. So the solutions have reached equilibrium on each side of the membrane. Here's another example. In the figure below, we have a glass or a beaker containing a sac holding a 70% sugar solution. The sac is permeable to water but not sugar, and the sac is made up of dialysis tubing, and it represents the plasma membrane in this case. So we often use this as an example of a cell model or an osmosis model mimicking a cell. So here are some questions for you. Where is the concentration of water the greatest? Is it in the sac or surrounding the sac? In which direction will water move? Into or out of the sac? Will the sac swell or will it shrink? Let's see if we can answer those questions together. Well, 100% water has a higher concentration than the 30% water that's in the sac. So water is in a greater concentration surrounding the sac. Therefore, water will move from where it's high in concentration to where it's low. So it'll move across that membrane into the sac. And because it does, the sac will swell. And we can visually see that swelling, and we could also measure it by taking the weight of the sac before it was put into the beaker and after osmosis occurred. So let's dig a little deeper on these concepts of solute versus solvent and begin to use the terms isotonic, hypotonic, and hypertonic.
Osmosis and tonicity are often difficult concepts for introductory biology students to grasp. Tonicity represents the relative measure of dissolved particles or solutes in a solution. It is important to recognize that the higher the tonicity, the greater the concentration of solutes in the solution, and therefore the lower the concentration of water. Thus, a solution with 25% sugar contains 75% water. Here, we see a common lab experiment in which a bag containing 25% sugar is placed in a solution of pure or 100% water. The contents of the beaker have a lower solute concentration and higher concentration of water than the contents within the bag. Over time, water diffuses from the higher concentration in the beaker to the lower concentration inside the bag, causing the bag to swell. In general, water diffuses away from hypotonic solutions. Therefore, we can say that the beaker is hypotonic, which means lower solute in relation to the bag. In this experiment, water may move across the membrane of the bag, but not the sugar. In the next experiment, the solute concentrations of the beaker and the interior of the bag are the same. The term for this condition is isotonic. Iso means the same, and since these solutions are the same, over time we do not expect any net movement of water, and the bag should remain approximately the same size and weight. In the final experiment, watch what happens when a bag containing 25% sugar is placed in a beaker containing 50% sugar. In this case, the solute concentration of the beaker is higher than that of the bag. Since the concentration of water in the bag is now higher than that in the beaker, the water diffuses outward, causing the bag to shrink and lose weight. The term for this condition is hypertonic, with hyper meaning higher. There are some important conclusions you can learn from this experiment. First, notice what happened in both beakers A and C. In both cases, water moved from hypotonic to hypertonic solutions. The water, therefore, diffused toward areas of lower concentrations. Second, the terms hypertonic and hypotonic are always used in relation to one another. For example, in beaker C, we compare the hypotonic contents of the bag to the hypertonic contents of the beaker. Our bodies, and indeed all living organisms, regulate the movement of water by adjusting the solute concentrations in our cells. Therefore, an understanding of tonicity plays an important role in understanding how our cells and bodies function.
Okay, let's pick up with the idea, the concept, that organisms regulate water movement. Many organisms adjust the solute concentration in their cells by osmosis. This is called osmoregulation. In this figure, we see red blood cells representing animal cells and then a typical plant cell responding to solutions of different tonicity. In the first one, we have an isotonic solution, which will mean that the net movement of water will be equal going into the cell and coming out for both the animal cell and plant cells. Now for the animal cell, this is the normal condition. We want animal cells to be in isotonic solutions. But for plant cells, this is not the ideal. They would become flaccid in this case. In the second or the middle figures, we see the hypotonic solution. In this case, both animal and plant cells will gain water. Animal cells could lyse or break apart in hypotonic solutions. For red blood cells, we call this hemolysis. And in plant cells, they will gain water and become turgid, stiff. And this is actually the normal condition for plant cells, is to have turgidity, lots of water in the cells. They don't break apart because the cell wall maintains their shape. Unlike the animal cell, no cell wall, and the water will enter the cell and potentially break the cell itself. The hypertonic solution is the last case. Animal cells and plant cells will lose water in hypertonic solution. For the animal cell, it will shrink, and we call this crenation. And for the plant cells, it will shrink away from the cell wall, and we call this plasmolysis. And this is where we started at the beginning of this screencast. So I'm going to finish with just one more video on hemolysis and crenation of red blood cells, and I'll see you in class.
The image on the left is human erythrocytes, or red blood cells. The image on the right is enhanced with filters. These living cells are in isotonic solution. Note the lozenge shape. Now the solution has been changed to have a high salt concentration. It is hypertonic. The red blood cells shrivel and become crenated. Finally, the solution has been changed to plain water. The cells swell and burst, leaving empty membranes behind. This phenomenon is called hemolysis.